Well, good morning and welcome to the sixth and final session of the Bloomsbury Set Conference on Infectious Disease and Antimicrobial Resistance. I'm Ray Kent. I work for the Royal Veterinary College and I'm the lead person for the Bloomsbury Set programme. And it's me that will be chairing our session today. Just a few housekeeping notes before we get started on the session. Just to make you aware, the session is being recorded and it will be available on the Bloomsbury Set website after the event. As with other sessions in this conference, the format will be presentations followed by a panel discussion, and then there will be opportunity for questions and answers with the panelists. So we would encourage you please to post your questions in the chat box as we progress throughout this session. Don't need to leave it to the end. If you can put them along as you think of them, that would be great. And then we will select some of those questions to be put to the panelists in the latter half of this session. Okay. So we're on Friday's session. This is the last one in the conference, and I've been asked to present a short summary of the conference session so far. Well, hasn't it been fabulous? That's it. That's my summary. Okay, no, I'm teasing. I'd better give you a bit more than that because I think you'd be shortchanged otherwise. So I'm just going to run through a little bit about some of the key things I picked up from the week, and I've listened to all of the sessions, so I think it's been really interesting mix of things. On Monday, we started off the week with an introduction to the Bloomsbury Set and our conference themes. This was presented by my talented RVC colleague, Dr. Emma Tomlinson, who's chair of the Bloomsbury Set steering group. And Emma introduced two talks. The first one was by Jeremy Salt from GalvMed, and the second from Joyce Tate of the Inogen Institute, which is part of the University of Edinburgh. Their talks were focused on some of the main challenges associated with bringing animal health products to market, including reducing the financial risks and hurdles, and also the regulatory aspects of product development. The scale of the challenges as they outline them is clearly vast. And so the resources that we need to be brought to bear on these challenges have to be equally huge. But it's clear from the talks that we heard that patience and persistence do pay off. And that was exemplified, I think, by the story of how GalvMed has gone from strength to strength since its founding in 2008. On Tuesday, we moved along a bit to look at research translation in a bit more detail. Tuesday's session was chaired by the fabulous Dr. Alex Anderson of London School of Hygiene and Tropical Medicine. And this session was aimed specifically at early career researchers. So as I say, the theme was research translation, but rather than starting off looking in the life sciences, we heard a really fascinating talk from Chris Taylor, talking about something called SPARK, which is Cardiff University's new social science research park. Now SPARK, it's a bit like Jurassic Park, but with fewer dinosaurs. Fascinating collection of humanities and social scientists brought together to tackle interdisciplinary approach to tackling some really grand challenges in our time. We then moved back into the life sciences with a very lively presentation by Hugo Villanueva from Stevenage Bioscience Catalyst, and he described the work that they're supporting there in cell and gene therapy and the companies that are based on that site. Both of these talks were really excellent, and there was a very lively panel discussion afterwards, which contained some great hints and tips for researchers who are interested in taking forward their concepts to market. So if you're in that category, it's worth tuning into that session and listening to that. if you didn't hear it on Tuesday. Hugo and Chris both mentioned the importance of finding allies and mentors who can introduce you to the next steps in the process, and perhaps at some point make introductions to investors. On Wednesday, we moved to the topic of research funding. The session then was chaired by the awesome Dr. Eigen Kurt Dixon from the London School of Economics and Political Science. And as I say, the topic research funding, most important issue in our trade. There were 3 excellent talks. The first was from Helen Lambert of the University of Bristol. Helen is the UKRI Challenge Lead for Global Health. And she gave us a mind-blowing statistic that there's been nearly £6 billion worth of grants awarded since 2015 by UKRI and its predecessors in the area of infectious disease and antimicrobial resistance. So that's nearly £1 billion a year in that period. So we're clearly in the right space in terms of looking for grant funding. Helen's talk was followed by a presentation from Jo Frost of Innovate UK, but she was talking not about Innovate's programs, but about Horizon Europe, which is the next European flagship framework program, starting this year and running through to 2027. The day was rounded off then by a talk from Ruth Neal, who spoke of the Longitude Prize. The Longitude Prize was first awarded in the 18th century, the first winner being the watchmaker John Harrison for his marine chronometer. I don't know if you knew this, but I was reading up about this, and it took the board of Longitude 36 years to award him all of the prize money for his invention. And he was actually 80 years old by the time he got the last tranche of money. In those days, the Longitude Prize was worth £20,000. That's the equivalent of about 2 million in today's money. But it's been reinvented in recent years as an £8 million funding pot being offered by Nesta, which is the National Endowment for Science, Technology and the Arts. And their chosen area is antimicrobial resistance. Once again, there was a very lively discussion following the talks about funding and opportunities in this space. Well, that brings us on to Thursday, when we ran 2 separate sessions. In the morning, we had the incredible Caroline Broad, who is the Royal Veterinary College's entrepreneur in residence. She was chairing. There were 6 lightning talks based on projects funded by the Bloomsbury Set. These were quite an eclectic set of talks. They ranged far and wide, starting off by looking at the economics of tackling infectious disease and AMR. We moved on then to consider the use of artificial intelligence, machine learning techniques in combating AMR, and then switched to a very different topic, anti-helminthics. That's a hard one to say. These are drugs to expel parasitic worms and other internal parasites. And so we moved on to other fun things like antifungal nanoparticles, and finally ended with 2 talks on preventing disease and diagnosing disease in cattle. In Thursday's afternoon session, we considered innovation in the field of combating emerging infectious disease. In the chair this time was Dr. Ying Chen of SOAS, University of London. Ying is newly arrived on the Bloomsbury Set scene and is a very welcome addition to our steering group. The first of the afternoon talks came from Martin Smith of VMIC, the Vaccines Manufacturing and Innovation Centre in Oxford. He described how the centre is being established at Harwell and will have the capability of producing 70 million vaccine doses by the end of this year. The second speaker in that session was Sarah Cordery, the Global AMR Innovation Fund, GAMRIF. And she gave us another eye-watering figure that it typically costs $1.7 billion to bring a novel antibiotic to market. So we can see that the challenge is there. The final talk of the day was by Paul Davis of MoLogic. Thanks to Ying, I now know that Paul is the inventor of the Clear Blue pregnancy test. But actually, he was here to give us a fascinating presentation on a novel, low-cost, high-sensitivity lateral flow testing platform. We all know what lateral flow tests are now, thanks to COVID-19, of course. This particular platform has the advantage it can be deployed early to support early detection of tropical diseases, and therefore something that could be really useful in low and medium income countries. Well, pause for breath, phew. After that roller coaster ride through the conference so far, it's time for me to go and lie down in a darkened room while you listen to our first proper speaker for today. Now, it gives me great pleasure to introduce Janet Hemingway. Janet is Professor of Vector Biology and Chair in Insect Molecular Biology at the Liverpool School of Tropical Medicine. And she's going to talk to us about ICON, the Infection Innovation Consortium. So over to you, Janet.
Thank you very much, Ray, and good morning, everybody. Absolutely delighted to be able to join the Bloomsbury Set conference here, and hopefully the start of a longer-term relationship between the initiative that I'm going to talk about today and the Bloomsbury Set going forward. So ICON is something that has been about 2 years in the making, but formally started in September of last year. And it's a great example of interdisciplinary collaborative working across a huge range of different disciplines. So I hate to say what's the start of those disciplines and what's the end, but we've got everything from engineers to social science, to lab people, to clinicians, to people working in big data, modeling, etc., etc., etc. The list goes on and on. Even though we've only been up and running for several months, we already have about 150 people working in this program. And that's just the people directly employed on the projects within it. If you add on the collaborative people working on it within our industrial partners, it's much more than that. And by the time we get fully up to speed, hopefully in about 2 or 3 months' time, we should be up to around 500 people working on this large-scale program. Next slide, please. So what are we actually doing? We're actually looking at the anti-infectives landscape. And by anti-infectives, I mean everything from drugs, antibiotics, public health insecticides, diagnostics, and working out where some of those pinch points are in getting things through to market. Next slide, please. If you actually look at all of those different anti-infective therapeutics, they all have similar characteristics. It doesn't matter whether you're looking at an antibiotic or a vaccine or a drug or a public health insecticide, you're operating in an underinvested market, which is in need of new solutions and innovations, where innovations primarily come from research institutes and small and medium-sized companies, but we're a large industrial partner to actually take many of these things through to the market. Where the markets themselves tend to be dominated by products that are at the end of their life cycle, and where you're working in relatively high risk space. So you've got lots of products or lots of molecules coming in at the bottom end of the pipeline to get one molecule out at the other end of the pipeline. And you've already heard Ray give you the figure for an antibiotic. We could give similar eye-watering figures for vaccines or public health insecticides or even diagnostics to actually get these through to the market. So what we wanted to do with the ICON consortium was really to try and enable innovation, to try and get over some of those common roadblocks that were in the program, to de-risk development by working closely with our industrial partners, facilitate that work in a cheaper and quicker format than we do at the moment. And I think what you've seen with the vaccine development from COVID shows what can be done when people really concentrate on trying to shorten safely that pathway of getting things to market. and make sure as we did this that we developed platforms that were offered to everybody in industry, because we want these to become the new normal in terms of the regulatory pathways and getting things through to market. Next slide, please. So to do this, you're not looking at a small program. We had the core of our funding come through the UKRI Strength in Places program. So we won 18.6 million from that program. We have 6.6 million from the European Union, and then another 135 million from the various collaborative partners who are working with us. So a total program, when I made these slides a couple of months ago, of 160 million. In fact, this is growing exponentially at the moment, and so we're now up to 175 million, even from the time that I sent these slides through to Ray and his team to put into the program here. Next slide, please. We've actually anchored the program in the north of England, specifically because it came through the Strength in Places program. But what it is 6 physical platforms and 2 virtual platforms that look at everything from early stage development to late stage getting the products into market and making sure that they're properly positioned in the marketplace to get the best benefit in terms of the market size for the company taking them to the market, but also to get the greatest impact in terms of the health benefit we're seeing. So what I wanted to do is just give you a couple of examples. I don't have time to go through everything in the time here. A couple of examples of how we think that might work. Next slide, please. So we're looking really at enabling through partnership small companies working with the research institutes and larger scale industry to actually take things through from the early stage discovery. We're co-developing things with the industrial partners in different formats, and it really depends on what stage that the partner is at when they come into the program as to how that works, but it's very flexible. We're validating where those products should be in the market and looking at the market opportunities for companies as they go through. And then making sure that when companies do have a product ready to go into market, whether that's in the UK or into global markets, that we help them get the best positioning. Next slide, please. The aim is not small. We're actually aiming to become the first choice for an anti-infective translational research for many, many companies working with us, and to move the dial. So if you look at anti-infective R&D in the northwest of the UK, it's currently worth about £2 billion a year. We're hoping in the next 5 to 10 years to turn that sustainably into at least £3 billion a year. So a step change from where we already are from quite a high baseline. Next slide, please. We're going to do that in a whole raft of different ways, but it includes actually generating new spin-outs for companies who are working in niche markets, and we have 3 of those already planned that will be soft spin-outs from the university systems. To look at radically innovating around product development in a whole range of areas. And I've already mentioned the regional economic development that we look to see from that. Next slide, please. So what is the offering? Well, I think the best way of trying to describe this is to give you 2 or 3 very quick examples of what we're doing, which will also give you an idea of the sort of breadth of the activities that we're starting to cover. So of the 9 circles that you've got on here, each of these represents either one of the physical platforms or one of the virtual platforms. And then the 9th circle is actually a specific fund that is there for us to work with small and medium-sized companies who are wanting early stage advice in terms of how they take their products through and how they might access the rest of the platforms that we've got. Next slide, please. So if I take a specific example, which is really very relevant to the conference here, it's around antimicrobial resistance. And the platforms that I've got highlighted there in red are all relevant to the antimicrobial resistance. So you could access our program at the very early stage where you're looking at actually the discovery of new molecules, right the way through to the product placements. Next slide, please. So one of the early stages that we've got is actually giving access to companies a brand new natural product library that we have. This came about through an initiative that Adam Roberts within the Liverpool School has been generating with a swab and send initiative of citizen science, where he's basically asked anybody who wants to get involved to take a swab of something. We don't really care what it is. We don't know where we're going to find bugs that are actually producing new antibiotics. So this is a really random selection of where they're coming from. Those swabs are sent back to us, and we grow whatever comes back on those swabs on baseline agar, but also on a diverse range of different plates that allows us to see whether those isolates then are producing anything that is of real interest. And you can see on the slide that we have 1 or 2 isolates there that are producing things that kill, for example, Candida albicans, which is a multi-resistant Candida albicans. So clearly something interesting that is being produced by that isolate. We've teamed up with AstraZeneca and Unilever and are now fractionating that huge natural product library, which is literally hundreds of thousands of isolates. And we'll be able to offer that as a mass screening system on the AstraZeneca industrial platform for screening of new products. It'll be set up as a modular library. So if companies want to come in and particularly want something out of soil or skin or home isolate libraries, we can do that. But they can also screen the whole library for new molecules of interest. And we've got a lot of interest from industry wanting to come through and look at this particular library. So that's on one end of the spectrum where we're looking at discovering brand new molecules. Next slide, please. But on the very other end of the spectrum, we're then trying to work out where the best placement is for some of these products. And these are not necessarily antibiotics that you're going to give to people, but by looking at 3 different countries, Malawi, Uganda, and the UK, we're trying to work out where antimicrobial resistance gets into the environment and how it spreads. And we're doing the same with COVID right now as well. And if we understand where the hotspots are for shedding of these things into the environment, we can also work out where the best placement is for new hygiene products, for example, that we can put into the market that is going to reduce that environmental contamination and environmental spread. Next slide, please. So just to summarize for the antimicrobial resistance, we have activity that is on the develop side, using the massive natural product libraries that we will have available for high throughput screening. We have an established hits to leads program that we're running with e-partner Infex, and organizations like LifeArc and others are already working with us on that and bringing forward compounds that they want to take through to the phase 1 clinical trial stage. And we then have access to the phase 1 clinical trials unit that we operate collaboratively with the NHS Trust in Liverpool. We're putting stages in between the phase 1 clinical trials and those early stage developments to try and allow companies to fail early if they're going to fail with the compounds coming through. And that's providing new humanized organoid models for many of these things so that we can look at how these behave, not just on agar plates, but on things that mimic human liver, lungs, kidney, heart, things like that. We're producing new rabbit and mouse models so that we can get the most highly resistant antimicrobial resistant pathogens into a format where we can look at the pharmacodynamics and pharmacokinetics of the new compounds that are coming through specifically to target these. We're then providing a human challenge model for looking at early stage clinical testing of these things, and a range of different programs being set up there with high throughput screening activities that will allow us to do that. We're defining the way that these things are moving through populations, which is the final stage that I just mentioned to you. And we also have a set of modelers working with us so that we can actually model the movement and look at the impact that we believe that we're going to have with some of the interventions that we're putting in place. So an enormous spectrum of activity there that companies can come in at any one point, or may come through very early in that pathway of development and collaboratively work with us moving through that scheme as they go along. Next slide, please. Alongside the AMR, we're also looking at parasitic diseases and looking specifically there at parasitic diseases where it's really quite hard to do some of the early screening in a way that is really meaningful, particularly where you don't have a good animal model of the disease that you're working with. And so we've teamed up with various groups there to look at human organoid screening and drug optimization, where that's needed in Cat3 facilities, where it doesn't need Cat3 facilities, that will be operating in Cat2 facilities. Some of those early stage development, but taking those all the way through to being able to offer them as a screening platform then in a specialist contract research organization type function. Next slide, please. The human challenge models are something that I think we've been at the forefront of for quite some time now. And we've been developing those for pneumonia, but we're also looking at expanding the offering that we have there for RSV, influenza, multi-drug resistant TB, and we will be working with COVID as well in a human challenge format very shortly. Next slide, please. As Ray mentioned, when I was first introduced, my background is actually as an entomologist, geneticist, biochemist, jack of all trades, master of none. But I couldn't put a program like this together and have nothing on the vector control side. I've already established a product development partnership for developing new insecticides, which is still based in Liverpool, and that will interface with us. That's the IVCC program. But what we wanted to do for the vector control products was actually look at how we speed up getting those new products through to market. Because it's actually a very difficult regulatory pathway once you've got your brand new insecticides to actually get those into products that get the right approvals to take through to market. So we've teamed up with the Bill and Melinda Gates Foundation, the Global Fund Against Malaria Foundation and others, and we're looking at much better ways of doing randomized control trials that should shave somewhere between 5 and 10 years off the timescale of getting some of these products through to market. If we can actually prove that we can do this in a way that the regulatory systems will accept and take through, I think there may be things that we could do looking at the drug and vaccine regulatory pathways going forward as well. Next slide, please. Diagnostics is clearly another area that has been very high on everybody's agenda with the recent testing activities for COVID. And I should say that we put this bid into UKRI well before there was a COVID pandemic and anybody was worrying about this. But we have been able to pivot a fair amount of what we're doing so that we can work with companies who are developing new COVID diagnostics. We've worked with many companies, including Mologic, who gave one of the earlier talks within the consortium meeting this week, and helped those companies develop their testing, validate their testing, and we can take those through to MHRA approval. Given that we're a fine centre, we're a WHO centre, and have the right links to be able to take those through. But clearly, we're not just working on COVID, we're working on a whole raft of other diagnostics as well, and not just on the classical molecular diagnostics and lateral flow type tests, but we've got a whole range of engineers involved that will allow us to work with brand new sensor technology to be able to look at different diagnostics. Next slide, please. And so what we're doing with the non-invasive sensor diagnostics are two things. One is using that technology to measure insecticides on walls, for example, for a quality control system for new insecticides. And that new product is about to go out into the market in India within the next 6 months. But we're also looking at ways of measuring microfilaria, for example, in peripheral blood without needing to take a blood sample, particularly to support the antifilaria sort of program to try and eradicate that disease, where you a better monitoring system than we have at the moment. That means you have to take blood samples from people late at night. Next slide, please. So as I've already said, over the last few months after we've been going, we spent quite a lot of time working on SARS and looking at what's going on within that sort of program. And that has allowed us to get people from all sorts of disciplines working across the spectrum on that. So we've been the Northern Testing Centre for the AstraZeneca Oxford vaccine. We have worked with a huge number of companies to get the lateral flow tests through. We have been working with many of the hygiene companies looking at how their products will remove COVID from surfaces and things like that. And we've also done the environmental modelling for the UK in terms of how COVID is moving through sewage systems and other environmental systems. I think that gives you an idea of how quickly we can respond to need, but also how we're trying to structure this programme in a way that is very translational, but also gets things out into the market very quickly and allows us to work collaboratively with a vast range of partners to be able to do that. Next slide, please. So now we're up and running. We're clearly already talking to people in the Bloomsbury set about how we interface with what you're doing and how we can work collaboratively to improve everybody's programs that way. And we're actively looking across a range of areas for new partnerships and R&D collaborations where people can actually benefit from the technologies that we can bring to the table, because we want this to work for everybody. And truly generate an ecosystem within the UK and more broadly that is going to speed up our ability to be able to deal with infectious diseases and do that in a way that we've got all of the skill bases that our multiple organisations and institutions have that can work better in partnership than they do in their individual silos. So thank you everybody for your attention, and I believe I pass back to Ray now.
Thank you, Janet. That was a really fascinating talk. Lots to take on board there. Lots of really interesting things to hear about there in terms of where your program is going. Okay, we're going to move on now to our second speaker for this morning. I shall give you an imaginary drum roll. The speaker is Claire Heffernan. Claire is the Professor of International Development and Director of the London International Development Centre, LIDC. Her talk is going to consider some of the interdisciplinary approaches to grand challenges around antimicrobial resistance, climate change, and emerging infectious diseases. So, without further ado, over to you, Claire.
I'm chatting away. Yeah, I guess what I'd like to do this morning is take us through some of the big things that we've discussed this week, link that to actually implementing interdisciplinary research, and then I thought I'd present a little bit about one of the projects that I've been involved in the Action Against Stunting Hub. So I get to present on interdisciplinarity quite a lot. And whenever I do this, I also speak about wicked problems because they tend to go hand in hand. And I think our three global challenges that we've been talking about this week are clearly wicked problems. And this notion of a wicked problem was offered way back in the late 60s and 1970s. Next slide, please. By these two researchers that worked I think they were at Berkeley, and they were urban planners, and they worked in India. And what they came up with was they looked at the amount of knowledge that universities and research institutes were producing. And then they declared that normal scientific processes were really only suitable for tame problems. And those are problems with readily identifiable, really discrete boundaries and a recognizable endpoint. And they thought that this wasn't really very accurate in terms of describing what's happening in the world. You know, the world is filled with very complex and intractable problems, you know, from poverty to environmental destruction to infectious diseases. And this was what they were thinking at the time. So the founding premise of a wicked problem is that it's so unstructured and complex that any attempt at resolution tends to flounder in a new set of problems. Next slide, please. And this was incredibly radical at the time. And they came up with a whole variety of parameters to describe a wicked problem. But I would argue that what these brilliant academics failed to see was that in 50 years, we'd be faced with multiple interrelated wicked problems all at the same time. Now, next slide, please. I wrote a paper a few years ago, and I argued that climate change was the new normal, and therefore essentially represented a baseline in which emerging and re-emerging infectious diseases played out on. And now I think we need to add AMR to this mix. Next slide, please. And of course, the interrelatedness between AMR, climate change, and emerging infectious disease means that new challenges are likely to emerge, such as multiple emerging infectious diseases. And this brings us, of course, to the current pandemic. Next slide, please. Now, you know, we know that COVID has had an impact on some of the most vulnerable communities globally, and all the evidence is not yet in. But we also know that this pandemic is not existing in isolation. Next slide, please. And take, for example, India. Now, at present, India has the world's largest population of stunted children. So of the 142 million children globally that are classified as stunted, the majority, 44 million, the highest concentration, are in India. And you see different now I've seen some new statistics that there's 60 million stunted children in India. There's another one, 54 million stunted children. So it shows you that this is a very, very large population. Now, we don't know about how this pandemic is going to play out on nutritional insecurity on these children over the next decade. You know, we don't have the evidence for any of that. And even some of the forecasting to me seems not quite right. The modeling, I think we've still got to develop. But what we do know, we know that these children live in very, very unstable environments. Climate change is really starting to impact agricultural production in India. But equally problematic to that, sort of cheek and jowl to what's happening with climate, agricultural production, food security in India, is India is the second largest global producer of antibiotics, which I'm sure all of you know that. And antibiotic pollution in the rivers and streams and waterways is increasingly recognized in India as an emerging crisis, effectively. I think 60,000 newborns a year die from drug-resistant infections. Now, the government has recently legislated against pollution from pharmaceutical companies, and of course, this is a very positive move. But it's clear that this is only the first step in addressing this particular problem. So, and in next slide, please. So what's the solution? Like, we have to unpack these wicked problems. And so we need to start from this point of unpacking complexity. Next slide, please. And the only tool that we have in our toolbox at the moment is interdisciplinarity. And of course, we've talked about this a lot this week, but one of the most interesting things about interdisciplinarity is how over the years, the concept has expanded. You know, now in the 1990s, you know, everybody was talking about multidisciplinarity. Now we might be talking about pluridisciplinarity, metadisciplinarity, transdisciplinarity, infradisciplinarity, cross-disciplinarity. And what it shows you is the concept itself is very unstable. So we're always seeking the next best descriptor. And I think at its heart, interdisciplinarity is about combining knowledge forms. And what all these different sort of labels show us is that you can combine knowledge in many, many different ways. So, but at each one of these, the sum is greater than the parts. Now, next slide, please. Now, this is just a very quick scan of PubMed, and you can see this is the number of interdisciplinary articles has gone up dramatically. But there's two things that this shows. One, it shows that academic interest, but we know that academic interest is always driven by funding. I mean, and I think that we can, you know, I think that's a truism that we can all admit. So we know that the future for academia really is in terms of interdisciplinarity. But when we start to look at the body of work, particularly in relation to AMR, climate change, and infectious disease, next slide, please. These are dominated by assessments of risk. So we're using this very broad-based tool to unpack complexity on a single dimension of complex problems. So it could be argued that this application of the tool is the biggest barrier rather than interdisciplinarity itself. And I would argue that in order to reach a solution, we need to use interdisciplinary research in a much more inclusive manner. And this brings me to the actual types of interdisciplinary research. Next slide, please. So within the literature, there's three broad types. There's instrumental, critical, and cognitive. And instrumental is largely about what we've been talking about this week. It's really problem-led, and it's pragmatic. It borrows methodological approaches across disciplines. The second type of interdisciplinary research, a critical, is much more epistemological. And then finally, the third one is cognitive interdisciplinarity. It builds on integration by looking at researcher dissonance. I mean, so it comes from the perspective of the researcher. Now, when you go to look at the literature, it's quite thin on these types of interdisciplinarity. I mean, don't get me wrong, there's not a lot out there. But when we reach further down into the literature to look for these details of project implementation, sort of the nuts and bolts of how do we deliver one of these projects, the evidence base unfortunately gets even thinner. So next slide, please. So what we have is best practice guidance is exceedingly rare. And there is a lot less academic interest in the sort of design, management, or evaluation of interdisciplinary projects. So I thought I would just offer a little bit of personal experience here. So I think the design of interdisciplinary projects falls into two broad categories. Next slide, please. And the first, I always term this as this convergent knowledge. So it's really where one related question builds off another. And these projects tend to combine the building blocks of a wider body of knowledge. And it is these projects, anyway, that in my opinion have a much easier time of moving toward a solution, or at minimum, a more complete understanding. Now, the second type of project design is what I term endgame. And here, interdisciplinarity really combines data and builds outputs that occur at the end of the project cycle. And this is the most common type of design, the most problematic, and the most difficult. It's very hard to make connections at the end rather than putting them in place at the beginning. Next slide, please. Now, the other element of interdisciplinarity that we always talk about is researchers' qualities. And these are kind of very well accepted, that people who are good at interdisciplinary research tend to have a flexibility around thinking. And what I like is that a thick skin, because I think that is actually true. And this is where sort of the field of cognitive interdisciplinarity is so interesting, of using a cognitive science lens to explore how disciplines and knowledge outputs can be integrated. Now, in the final bit of my talk, I thought I would present a little bit about the Action Against Stunting Hub. Next slide, please. Now, we are a consortium of 18 institutions working across 3 continents, and to try to get into the solution space regarding child stunting. So who are we? Next slide, please. We're about 145 people, and they range from, senior academics to postdocs to research fellows to as research assistants to lab assistants, field staff, enumerators. There's a whole variety of people that we comprise. So, and we have 2 very ambitious aims for this hub. Thank you. In relation to interdisciplinarity. So the first, we wanted to identify the constellation of features and factors where child stunting can be ameliorated or potentially reversed. And equally, we want to know when is the best time to intervene. So how are we going to do this? Next slide, please. We designed the hub around what's termed the whole child approach. And this was first utilized in the education sector in the US. And basically it was decided that there was this real need to look at failing schools. And when they looked at failing schools, they couldn't identify one particular point in these schools or one particular element. They had to look at the whole child, really, to work out what was happening. And so we very much adopted this and applied it to stunting. So to explore this whole child, our researchers certainly cover a lot of disciplines. Next slide, please. But, in our paradigm, that the child is the agent of change. So we're looking at this nested ecology around the child. So we start with the physical child, and here we want to unpack sort of the deep biology from the epigenetics and the gut microbiome to pathogens and their interactions. These findings then link to the home environment that the child lives in, from diets across the first 1,000 days to behavioral messaging regarding wash, to complementary feeding, the purchasing of snack foods. And next, we're going to look at the educational environment. And here we're going to look at screening tools to develop cognitive deficits and assess caregiving behaviors and really identify what's best practice around these children. How can we best support them? And finally, we're going to map this wider food environment, including some of the market forces that drive the accessibility, affordability, and availability of food that this child will have access to. And next slide, please. And so we have a huge variety of different disciplines whose knowledge and expertise we need to integrate. So I thought I would take you through the three levels of integration. And the first is, next slide, please, is around the literature. So we're undertaking a whole big variety of reviews around the boundaries, explicitly at the boundaries where these different disciplines interact. So from the deep biology to the home environment, to the educational environment, food systems and interventions. And next slide, please. Second, in relation to our data. In order to be properly interdisciplinary, everybody has access to data. They need all of the data. So historically, it's very common that these large interdisciplinary projects really run like interdisciplinary projects run like a disciplinary project. Everybody goes off, takes their data, analyzes it, gets their disciplinary outputs, and then throws the data back into a big pot, and they see what happens with regard to interdisciplinarity. And I think that is a fundamental mistake. In order to be successful at interdisciplinary research and move into a solution space, you have to identify these nodes, these places where the different disciplines, where different data interact. And that's something that, we don't know a lot about. We're not very good at it. And a lot of people don't think about this when they're designing or planning or trying to implement one of these projects. And then the last element that I wanted to talk about, next slide, please, is how we're going to try to inform the field. And here we're going to try to add to new knowledge by looking at some of these elements of cognitive interdisciplinarity to identify and look at how well have we managed integration, and equally how well have we eroded disciplinary boundaries across this endeavor as we reach towards our outputs. So finally, I just had a final slide, last slide, please, just a few conclusions. And I think, good interdisciplinary research dissolves boundaries. And we have to look at that as we go through these projects. Best practice, we need a lot more about best practice. It seems like, we just reinvent the wheel at every project. And this is a huge waste of time. There's a huge amount of expertise out there. We need to be able to tap into this better. And finally, reaching back to the theme of this week, I think when we connect capabilities across these three grand challenges, these three wicked problems, we have to move beyond risk to actual outcomes. So I want to thank Bloomsbury Set for inviting me to present today. And I want to thank Ray and Emma and the rest of the team and Sarah. So thank you very much.
Thank you very much, Claire, for your excellent presentation. Can I just remind you, if you're out there in audience land, please put your questions for the panel discussion that's going to follow. We really want to hear from you. What are the key points that you would like to raise from the talks today? So please put those in the chat box, which you should be able to see on your screens. I'm going to move into to start the ball rolling with quite a general question. I think I'll put this to Janet first of all, if I may. Janet, these problems that we've been talking about seem so huge and intractable. Where do we start? How do you decide which interventions will make the biggest difference?
There's a difficult one. So I guess what we're trying to do is exactly what Claire's been talking about in terms of that interdisciplinary activity, to pull together all of the information that we've got coming from different areas, different strands, and then think about, okay, if we were to put an intervention in at this point, what would that do? We get modelers involved very early on in this process, and there's an iterative loop then between the modelers and the people who are collecting data on the ground to try and validate where we think the direction of travel is, and sometimes that points us in a very different direction. And the idea from what we're doing from that large-scale project that I talked about in Malawi, Uganda, and the UK, is to try and work out whether there are some generalities we can come through with here, or whether we're really going to have to look at this very differently in different high, middle, and low-income settings. But I think we're going to be surprised to the level of generality sometimes that you can come through within some of these areas, where we can learn clearly from different areas, and where there is a lot that would allow us to change that the way we think about how we intervene and when we intervene.
Thank you. Same question for Claire, if I may.
Okay, thanks, Ray. Can you just give me what was that again? Sorry, I lost it.
That's fine. So I said, so the problems that you've been talking about are kind of huge and seemingly intractable. So the question is, where do we start, and how do we make a decision where our intervention could make the biggest difference?
No, I mean, that is a really important question, because it becomes like, how long is a piece of string, and where do you start on that piece of string, and how do you know that you're at the right place? But when you're designing these interdisciplinary projects, you have to look at the problem itself, and you have to look at the complexity around the problem. And one of the things that I'm very interested in is called solutions-based research. And at solutions-based research, you start at the solution, right? Not at the problem, at the solution. And when you start at the solution and you start working back, it's subtle, but it's completely different. The kind of choices that you make in terms of developing a strategy to get to that solution. So that's kind of the way I see it, is that we have to start backwards. What does a solution look like to some of these challenges? And how can we create this new sort of architecture to deal with some of them? And I think there is a need for sort of radical and disruptive research, but putting it in a different kind of architecture that takes us to the solution. So I think we are looking at it. I think we've got to go the other way. So thank you for that question, Ray.
Okay, so start at the end and work backwards.
That's
from
right.
there.
Absolutely. Yeah.
if I can summarize it.
Yeah.
Okay, thank you. That's really interesting. Slightly different question for me again. So both of you, both panelists today have been incredibly successful when it comes to winning large grants. I think it might be interesting if we could if you could tell us what your secret is. I'll start this one with Janet, if I may.
What is the secret? I think being able to look at the big picture and then translate that big picture into something that is meaningful, not only for academia, but also for the other stakeholders that are absolutely equally as important within that breadth. So it is being able to take a problem and say, if I'm going to pull a group together here that needs to involve the NHS, the Environmental Agency, Public Health England, and large and small companies, how do you actually set that up in a way that everybody understands what it is that you're trying to do and how you're trying to do it, and understands how they might actually contribute to that? And I think if you can get that big picture strategic thinking right, then it's relatively straightforward to sort out the smaller bits. Often I find people go the other way, and then they've got their own little pet project and they try and work out how they can grow that into something bigger. And I don't think it works that way particularly well. But it's doing big strategic thinking up front and then working out how you place things in that framework.
Okay, thank you. I guess this is the direction of travel has been for some years now in terms of particularly UKRI funding is becoming much lumpier, if you like. So same question for you, Claire. You have the Action Against Child Stunting Hub, which is a grant, I think, similar size to Janet's, about £18 million worth of funding over 5 years. So what's your secret?
I think when you said that, Jenna, I absolutely agree with you. I mean, what you have to do is you have to look at a body of work and look for the gaps. And to me, that's what I do. I lean into a body of work. I look for the gaps, and I look for the solution space. And then I put a team together that I think can deliver that solution. And that's what we try to do with the Action Against Stunting Hub. So, I mean, knock on wood, hopefully we'll move forward into what has been, an intractable problem and make headway against that. And that's, part of being an interdisciplinarian. I think it gives you a bit of an advantage to do that. But you're looking for gaps and then how you can fill them. That's a trade secret. I don't know if I should give that up.
That makes good sense. Okay, we've got some really good questions coming in from our audience today. So I'm going to put a couple of those to you, if I may. First one is a question for Janet, and it's: how should universities seek out their commercial partners? Should they focus on academic relationships with companies, or presenting new companies to academics, or a combination of both those things?
Okay. So I guess I wouldn't do it either way. I think to me, it's crazy that academia and industry doesn't work more closely together, and they should be doing and could be doing even more than they already are. But I think, again, it's thinking from that bigger picture start point. So if you're coming at it from the academic side, from the university hub, what is your sweet spot in terms of what have you got to offer to a partnership that is going to take something forward, that's going to take your agenda forward and industry's agenda forward? And once you've worked out where your skill sets are, then it's talking to the right industry people at the right level in the right language. And often what happens is that I think academia goes with just an area of its own interest and doesn't present it in the right way that they're going to engage industry. They talk in the wrong language, they talk in the wrong time frame, they go asking industry to fund everything but do it at the wrong time within that product development cycle as well. So I think it's really concentrating quite hard on what is that sweet spot, and then growing long-term collaborative partnerships. So my first industrial partnership I set up with a big multinational company back in 1981, and I'm still working with that company, and that company was part of my IVC program and is part of this program. That's an industrial collaboration that works, and it works because both parties get a lot out of it. And you only need a few of those, and other companies want to come in and join that party as well because they can see how well it works. But beware of just going with an academic idea or speaking too academically to the companies, because it will tend to go right past them if you're not very careful.
Okay, that's really interesting. So I guess you're saying there that a long-term relationship can be established if you hit the right sort of sweet spot, as it were, which is clearly desirable. Okay, thank you. Another audience question, this one for Claire. So this is a question around that we've hinted at the scale of your program, your GCRF hub. You mentioned that you had around about 150 researchers from different countries, different disciplines. Mm-hmm. When you're managing something at that scale with different languages, different cultural expectations, different personalities, human beings, some will get on, some will not get on. How do you ensure equitable partnerships, and not just in a north-south kind of context, but generally?
Yeah, no, thank you. That's a great question. And it's something that we really thought about when we designed the hub. So what we did was we created this very flat management structure. So every post in the hub that's is mirrored in the country hubs. So for example, we have a senior hub project manager in the UK. There's an equal post in India, Senegal, and Indonesia. So there's a data manager in the UK, there's an equal post across. So there's a theme lead from maybe a UK institution that is going to be a theme lead in each of the countries. So what we've done is we've created these sort of cohorts of equality, essentially. So the senior hub manager in the UK has the same job that the senior hub manager in India does. So there's a lot of sharing of knowledge. So yeah, equitable partnerships is one of these really challenging things. And one of the things that by this flat management structure, it's very flat. So there can be problems with that. But what we tried to do was to create sort of equality from the ground up, and that there would be no difference in anybody's position. And we also did a lot around ensuring that our executive board has representation from the country leads, and everything is kind of an equal endeavor. It takes a lot longer. It's a much more complex dynamic. Decision making is not like a quick thing. But you know what? I think that's okay. I think we get there in the end, and we'll see if how this design fares in terms of forging equitable partnerships. We'll only know that at the end.
Okay, thank you. That's a really good and interesting answer. Just a reminder to our audience, please keep your questions coming in. We'd love to have more questions from you, please. So just linking into that question again, another one for Claire here. And again, it's just thinking about sort of equitable partnerships. How will your team integrate the findings from the communities that you're working with at the end of the project at a level that is appropriate for their understanding of the issues? And do you have any concerns about that?
We have a large public engagement component, and we also have a citizen science component. And so we have a lot of different, and we also have a shared values component, where we identified community level share values, and then we look at the legacy over time and how these changed, and how the project in any way might have influenced those. So we've really thought about that when you do this kind of research, we've got an observational cohort and we're taking lots of blood samples. It can be very extractive by its very nature. And how do you soften that to ensure that you're engaging communities appropriately, and that you're going on this journey together? So it's not easy, but we've done the best we can in terms of engagement activities. And what we found is one of the problems in the field is more women want to be part of the project, and it creates a bit of that, it's a little bit of, we have to be careful of the social dynamics, because apparently it's quite popular in many countries, particularly Indonesia. So yeah, so it's been an interesting journey, but it is something that we have to be very, very, very aware of, that our footprint needs to be light, and that we need to give something back to these communities.
Okay, thank you. There's a question now that I think I'd like both panelists to think about. So this is a question about interdisciplinarity, but the question is, how can interdisciplinarity help us to stop antimicrobial resistance from becoming a global health catastrophe of the same or similar proportions to COVID-19? Perhaps if we start with Janet, please.
Yeah, so there isn't one solution on this, and we're going to have to tackle this at all sorts of different points within that sort of spectrum of feeding the pipeline with new things, but that doesn't answer the question, because you will lose things quicker than you can feed them in to the system. Stewardship of what we've got, but making sure at the same time that you can actually treat people and keep people safe. And so there is a whole range of different areas there that we need to look at. What are we bringing in that is new? How do you introduce that, and how do you get the best stewardship of those products as you bring them into the market, so that you keep them there and you keep them effective for as long as you possibly can, and get the greatest public health benefit out of them? And sometimes there's a trade-off on those things, but it's getting people involved right at the beginning with those discussions. And we've been doing a similar sort of thing with the insecticides, because public health insecticides were probably in a worse position, to be honest, than the antibiotics, where we were running out of things and there was nothing in the pipeline. So we've re-stimulated the pipeline, but knowing that pipeline is incredibly thin, actually getting agreement up front by all of the companies we were working with that they were going to work with us on the stewardship of those compounds and how they were brought to the market, the format they were brought to the market, and how they were going to be used to try and keep them there and keep them effective for as long as possible. And a lot of people thought we wouldn't be able to do that because we were going to have to talk to different companies and get different companies to agree to collaborate and share a market in the way that market forces often don't allow you to do. But it's been interesting to see that they understand it's in their interest to do that. And we do have companies talking in a way that I think a lot of people thought 5 or 6 years ago just wasn't going to be possible. So maybe we can do the same in the AMR space.
Thank you. So Claire, do you have anything to add to that?
Yeah, it's interesting. With AMR, one of the things that we're very interested in is antibiotic pollution. So we have a project now in Kenya where we're working with engineers, public policy, microbiologists in terms of developing these low-cost banana leaf filters to filter out some of the antibiotic pollution in the Nairobi River, which is one of the most polluted rivers in the world. So I think that the AMR solution will be largely technical, and I think that we're going to have to really combat this environmental aspect of this. And I think that we need low-cost solutions, sustainable solutions for the global South. So that's what we're working on at the moment.
Thank you. Audience, please do keep your questions coming in. We've got a number still to answer, but there's be great to have some more, please. Okay, it's a question again, I think both panelists might want to dip into this one. It's how can we encourage academics to join or engage in translational programs? How do we present the value of these activities and the benefits to them, considering conflicts such as confidentiality and the ever-present pressure to publish your work? Who'd like to answer that one?
I'm happy to make a comment on that because it's clearly a space that we all work in when you're in these sort of areas. So what I say to everybody on this is that as an academic, not everybody needs to be doing things that are immediately translational, but those of us who are working in the space that believe that should be the case, you get a huge amount of satisfaction out of it if you actually see your research being taken up, being put into a product that gets out there and actually saves lives, because that is really what drives a lot of us working in this infection space. And B, it doesn't mean that you can't publish if you're working in this sort of space. What it means is that you can't potentially publish at the same speed as you might, but once you've got a portfolio of activity set up, then you can still publish as long as you've got things properly patented, as long as you've got things properly covered. And so, as somebody who works in the translational space, I still turn out around 8 or 9 papers every year, and still hit Science and Nature and PNAS. So it doesn't stop you doing that. You just need to do it in a way that actually doesn't stop you doing the translational activity. But the satisfaction that you can get out of actually getting this right is enormous. So I would encourage everybody to think about how they can really get an impact out of their work by working collaboratively and not believing that they have to do it all themselves.
So Claire, do you think we need better incentive or reward structures within academia to encourage researchers to be more translational in their thinking?
I mean, absolutely. I think university structures aren't necessarily set up for that. And that's something, and university departments equally tend to be, I mean, this is changing. But I mean, historically, when we looked at sort of interdisciplinarity, because this is something I still get when I go and present like about sort of the benefits of interdisciplinarity, occasionally I'll get comments like, I can't possibly have my graduate students, I can't encourage them to be interdisciplinary because they won't have a career. So you still get this kind of bias toward deep collaborative work with other partners, whether that's translational or it's interdisciplinary research. I think there's still that universities are often not set up, although this is changing, to accommodate the kind of diverse research needs. And as Janet said, the publication timeframes are longer. You're working with more partners, it's more complex. There's a whole variety of different things that have to kind of come into the decision-making around publication. Data transfer, even when you're working in these big groups, is also a very lengthy element that can often take much longer time than if it was just a straightforward project. So yeah, I think it's challenging, and I think that we need to get over these barriers, and universities need to support this kind of research on a much deeper and broader level.
Okay, that's interesting. It would be interesting to sort of see university reward structures evolving in some way that rewarded group work rather than individual effort. But maybe that's something that won't happen quickly, but it's everything at the moment is very much around individual incentivization and not collaboration, I think. Okay, another question come in from the audience here. This is another good one. It's about knowledge co-creation, and again, if I perhaps could start with Janet on this. How do you co-create knowledge with industry, with companies? And what kind of practices do you use to sort of do that kind of co-creation? For example, in your ICON project, how's that going to work?
Yeah, it is hugely variable depending on exactly what it is that we're going to do. But we would start off working out what the background and foreground knowledge are. So in any one project, we'd sit down in detail with all of the partners and say, okay, where are we starting from, and who is bringing what to the table, and who owns what right up front, so that we've got that very and we get a very clear legal agreement then in terms of what happens to that knowledge, and also what happens to what we create jointly, who owns it, who benefits from it, and so on. It is way easier to get sorted out right at the beginning than it is to try and do it retrospectively, and any company worth their salt would want to do that as well. And once we've got the framework there, we will then work collaboratively over time to actually take things forward. What we tend to do is also put in time points, usually at about every 6 months, where we will review how things are moving, where we've got to, whether anything different has happened within that sort of process as well, so that this is a live collaboration and it's monitored in real time. We don't just assume that if we're setting up a 5-year program, what we believed was going to happen on day one will have happened after 5 years. And so we can then modify that ownership, that co-collaboration, and where we take that and what we can offer more broadly as we go forward. But it really is done on a one-to-one basis. There isn't a one-size-fits-all contract that you could hand out that says this is what you do.
Yeah, and sort of from your perspective, Claire, I mean, again, knowledge co-creation is clearly very critical to what you're doing. What kind of approaches work in your experience with the communities you engage with?
Yeah, no, it's really interesting how we developed the hub. And I mentioned this about looking at shared values. And so we've applied this approach through, and this was from the University of Brighton, called the We Value approach, which identifies community-led values. And from that, we've built off the different elements, the different thematic areas of the hub. So we start with, we have an understanding what people think good education looks like. What does good education look like? What is that? What's the value towards, how do people think about that? And that's informed sort of our education component. Nutrition, what does good nutrition look like? If your child, what's a healthy child? What's a healthy non-stunted child? Like understanding kind of how sort of a perspective, their perspective, and trying to use that to map onto what we're doing. A lot of the Action Against Stunting Hub is about detailed sample taking. There's a huge from the deep biology. And the other thing that's been really, really important for us is that, this data is owned by the participants. It's not owned by us. It's owned by them. And they're giving us this privilege of using this data. And they have a right at absolutely any time to come back and look at that data, to ask for what it says, if there's any tests. And that is not something that's actually standard practice, apparently, despite that it should be. So there's a lot of things that we hopefully have done to ensure that we have longer term relationships with our communities, and that we're giving something back to them. But knowledge co-creation is very much that. It has to be designed, it has to be implicit. And in interdisciplinary terms, what we're doing is we're building on it.
How do you manage that where you're engaging with perhaps migrant communities or communities that are certainly not static in some sense? How do you overcome those difficulties? Is that a big issue in the stunting hub, for instance?
We have urban informal settlements in India, and we're working with communities there. And the dynamics of these communities are very, very different. And they're less migrant, in my experience, than in other countries, but they're less sort of socially tethered in some areas than in other, if they were more rural or peri-urban community. So and so, for example, one of the things we're doing an egg intervention. So in India, we're feeding children egg powder just to see if it impacts growth. And one of the things that's happening is in some of the communities within these slums and some families is eggs are not considered a good thing. They're considered a bad thing. So, we looked at working with communities, engaging children. Can we paint eggs? Can we, how can we change this egg dynamic? And so, and that's very different. It's not the whole community in the urban area we're working with. So it comes down to individual households. So when you're trying to work with these communities that are more diverse, some of these elements or values or understandings are very, very different between households, and you need to accommodate that. So that's one of the things that we're working on.
Okay, thank you. Another audience question. I think this was sort of hinted at in one of your presentations, which was around this danger of kind of not learning from what's gone before sometimes and reinventing the wheel or starting something new. I wonder, are there sort of best practice examples from really any country in the world that we can learn from to support translational research? Janet, have you come across some really good examples that you kind of inspire or draw upon in your work?
So I think the answer to that is yes, but the ecosystems tend to be very different. I mean, I'm familiar with Japan, India, and the US alongside the UK system. And those ecosystems are so different from each other that it's difficult to draw a generalization. But I would say that looking particularly at the difference between the US and the UK, where the UK does very well on that early stage innovation, but doesn't actually get through to the translation quite as well as the US does. And I think there are probably sort of three things that I would pull out there that I see changing, but potentially changing too slowly. One is mentorship, and really making sure that we get the academic base in the UK understanding what it needs to take things through and having the right kind of mentorship very early on, not waiting till somebody is in the late stage professional career before you start and do that. The other is the amount of venture capital that is available and the ease at which it can be accessed and the understanding of how you access it from the academic base, and I think that's still pretty poor from many universities. And the other is maybe from the university side. I think I've seen too many examples in the UK of what I would call trying to do hard spin outs too quickly. And so what happens then is that you get a spin out which is really one person and nothing else. It isn't the way that spin outs work sensibly. And if you look at the model that's worked in the US, and to an extent the model I think that has worked pretty well in places like Cambridge, is far more of a soft spin out, where the university nurtures a semi-spun out entity for a period of time to allow it to grow before it cuts the apron strings and turns it into something that has to completely stand on its own. And I think there, if we got that system working much better, and we got the understanding in the universities much better, we would probably succeed far better than we do at the moment. But if you just incentivize universities to count the number of SMEs they've spun out, then what you're going to have is a large number of spin outs and a large number of failures.
Yeah, which is a perennial issue, because that seems to be the thing that counts with some funders, numbers of spin outs rather than value and equity raised and all this kind of thing. Interesting. Okay, we're moving towards the end of the session, but I've got one final question that I'd like to ask to you both. And if we start with Claire, what advice, Claire, would you give to your younger self if you were starting out again in this area?
I don't know if I would change anything. I really don't. I started an interdisciplinary unit in Reading in 2000, and I worked with a philosopher, linguist, vets, social scientists, psychologist, a lawyer. And so I've had PhDs that run from computer science all the way through to linguistics. So it's been one of the most fascinating journeys so I don't know if I would change it. I would say, keep heart. Interdisciplinarity gets very popular, just hang in there for 20 years. But so I would say that it's been a journey, but I don't think I would change anything. But it's an interesting question.
Thank you. And same question for you, Janet. What advice would you give to your younger self?
Don't be afraid of taking risks. So I'm a risk taker anyway, but you learn as you get older that you can take really big risks as long as you've actually thought them through, and very often they come off. And if you don't take those risks, you would never know. I wish I'd learned that probably about 10 years earlier than I did, in terms of the scale of risk and the scale of vision that you could take forward and take people with you.
Thank you. That's a really good answer. Okay, thank you to both our panelists. I'm just going to wrap up now with a final word. You may have seen in the conference programme that the title of the session included the statement, some next steps for the Bloomsbury SET programme. So just a couple of words about that. Our current funding from Research England finishes at the end of June this year. But in September last year, we applied to Research England for a follow-on programme to our current activity, which would run from the 1st of July 2021 for two years. As was kind of hinted by Janet earlier on, the follow-on will allow us to join forces with the ICON consortium, creating a London-Liverpool alliance for tackling infectious disease and AMR, which we think would be really exciting. So the key question that's kind of on all your lips is have we been successful in that? Unfortunately, I can't tell you just yet, because the news on that is currently something that we're unable to talk about in any detail. But I will end the presentation to answer the question, have we been successful? I will not give away fully the answer, but my gestures might indicate where we might be going with that. Okay, fabulous. So thank you. I'd like to just close now. We've got a slide just to say, really to say thank you to everyone that's been involved in this conference. Just to remind you that the recordings of this session and all the other sessions during the week will be on the Bloomsbury Set website shortly. I'd really like to say thank you to the organising committee for this conference, especially to all of our speakers and to our session chairs, and to the amazing team that manages the Bloomsbury Set. Also, thank you to the team at Visair who provided our conference platform this week, and I hope that that's been something that you've engaged with and found to be very useful. And last but not least, to you, our wonderful audience, thank you for joining us. And we will be having a second conference later in the spring, and we'd encourage you to sign up to that once we release details about that. So for now, I'd just like to say thank you once again to everybody. Thank you for listening in to our sessions, for engaging with putting questions to our panelists. And please keep safe and God bless. Thank you.